Good morning, everybody. Thanks for joining. For those of you that are new to the Stripe Collaborative community, welcome. We're glad you're here. For those of you that have been participating with us for more than a few years now, great to see you back. We have a pretty action-packed meeting this morning. We do have a presentation from McKenna Tennant at the FDA, CDRH, that we're going to be getting to here shortly. And we're going to have a moderated Q&A along with that. Just a quick preface on that. There will be certain questions, I'm sure, or concerns that a lot of you guys have regarding LDTs. Those are going to be excluded from today's conversation, but there is a very specific meeting link or information request where you can send those questions directly to the FDA. And then we're also going to hear from Terry Klein later today as well. So with that, Sarah, let's get going.
Great. Well, first we'll go through some updates. We did want to mention that registration will be opening up soon for our first in-person Stripe meeting and consensus workshop. This will be held in October, on October 23rd through 25th. It's a Wednesday through Friday at USP campus in Rockville, Maryland. USP is known for working to improve global health through public standards in many areas of medical and pharmaceutical industries. An emerging area for USP standards development is pharmacogenomics. And this consensus workshop represents an important initial step and opportunity for our community to come together to inform the rigorous process of developing a USP standard for pharmacogenomics. And so this meeting will be a hybrid meeting. So it'll be there will be an option to register to attend virtual. There is going to be a cap on in-person attendance. It's going to be 250 max. So if you do want to attend the meeting in person, just make sure to register when we send the meeting link out, which we think should be in the next 2 to 3 weeks or so. We have 15 different health sectors and more than 150 organizations represented in the Stripe community. So the idea of holding a consensus workshop is to convene stakeholders from across all of the different sectors in the pharmacogenetics testing ecosystem to share perspectives and practices regarding a targeted set of issues to inform a set of consensus statements. So the format for this will be panel discussions in the mornings on the first 2 days, followed by workshops in the afternoons. There are tentatively five workshop topics, and attendees will be able to select which topic they'd like to participate in. The third day, we'll have presentations summarizing the workshop discussions with a live vote and feedback mechanism to provide supporting or dissenting statements on the consensus statements. Following the meeting, any consensus statements that are developed during the meeting will be made available and opened up for public comment. So by registering, you will agree to participate in a survey that will be disseminated in advance of the meeting to understand various perspectives on the topics that will be covered during the workshop. In addition, we'll also provide a briefing packet in advance of the meeting that we would like everyone to review beforehand to ensure that attendees are informed on the various topics that may be covered during the meeting, regardless of each person's specific background and expertise. So the priority area is approaches to evidence evaluation for gene-drug associations, and tentatively the workshop topics are clinical decision support, health equity, clinical challenges, laboratory challenges, and study design considerations. So a lot more information regarding that aspect of it will be provided in the coming weeks. So just wanted to say thank you to our Stripe Conference Organizing Committee. We are kind of beginning to build out the workshops and the pre-meeting materials, so there are a lot of opportunities to get involved. And please feel free to shoot us an email at stripe@stopadr.org if you'd like to participate in the planning process. So with that, I'll turn it over to Kristine to give us some updates on the Right Drug Dose Now Act.
Thanks. And I'm just going to remind everybody to mute if they haven't. Somebody had a rather annoying metronome. It sounded like clicking through the entire thing there. All right, well, I had a few slides, but I can just talk through them. So the Right Drug Dose Now Act of 2024 was reintroduced at the end of March by Eric Swalwell and Dan Crenshaw. So it is bipartisan. There is details on what is included in that at fourthcause.org/RightAct, but essentially updates the National Action Plan for Adverse Drug Event Prevention to include PGX within 180 days of enactment, includes education on pharmacogenomics for healthcare professionals, all sorts of healthcare professionals as a key component to reduce adverse drug events. Plan to improve each EHR systems to utilize pharmacogenetic information, including flagging for appropriate testing and drug-gene interactions, as well as gathering real-world evidence. A GAO study on FDA drug-gene labels, and an update to make it easier to report to the adverse drug event reporting system, including a patient-friendly submission option, automating reporting from EHRs. There is a separate bill that will be introduced later by Swalwell for authorized funding. It's currently sitting in the Energy and Commerce Subcommittee. There is, if you Google House Energy and Commerce Subcommittee, it will pull up a list. These are the people we really, really need to target if you're supportive of this and they happen to be in your area. We are looking for additional volunteers to lead outreach efforts in their states. This is not a hard process. It's essentially either calling or emailing to request a meeting with the representatives in your area and telling them why you support this legislation, especially helpful if you have impactful patient stories in your particular area. But we do have template outreach emails as well as template decks. If helpful, you're also welcome to create your own. But please email me, kristine.ashcraft@uscript.com, or you can go to fourthcause.org/RightAct to learn more about how you can support this and help. At a minimum, would love you to sign on your individual support if you have not already, or get your organization to sign on support. Again, fourthcause.org/RightAct.
Great. Thank you so much, Kristine. We do have an open discussion at the end, so we can save questions for the end as well, but we'll go ahead and move into our featured presentation. We have joining us McKenna Tennant. McKenna is a policy analyst for personalized medicine at FDA Center for Devices and Radiological Health. She's also CDRH's liaison to the STRIPE steering committee. McKenna holds a master's in public health from Yale, where she studied the history of genetic engineering. Welcome, McKenna.
Thank you. I'm going to take over. Is it possible to enable me to share my screen?
Yes, let's see here.
Thank you. It just says host disabled, participants sharing. And while that's getting set up, I would also echo the previous sentiments, if possible, to mute your mics if you're not chatting, just because there was some feedback.
All right, try now.
Okay. All right. Can everyone see my screen?
No.
Yes, ma'am.
Okay, great. Thank you. So I wanted to compliment my CDER colleagues' past presentation to this community on CDER's perspective on pharmacogenomics, and provide the community with CDRH's perspective, focusing primarily on the pre-market review of pharmacogenetic tests. And before diving in, I do want to start with a disclaimer that I have no relevant financial relationships pertaining to the content of this presentation, but I also, per the limitation of FDA liaisons' contributions to collaborative communities, wanted to provide the disclaimer that any comments made by myself and FDA liaisons generally during discussions within collaborative communities are not formal agency communications, and the comments represent the best judgment of the FDA liaison at the time, but don't necessarily represent the formal position of FDA. if any, and don't bind or otherwise obligate or commit the agency to the views expressed by FDA liaisons in a collaborative community. So any viewpoints or proposals or recommendations regarding regulatory policy, regulations, or legislation expressed by or within STRIPE don't represent the formal position of FDA. So again, the discussions from this collaborative community may help inform FDA's regulatory decision-making in other decisions, but in which case FDA would follow the required substantive and procedural requirements consistent with the law and agency priorities. Okay, so moving on from disclaimers, CDRH, or FDA's Center for Devices and Radiological Health, strives to put patients at the heart of what we do, with a vision that patients in the US have access to high quality, safe and effective medical devices of public health importance. So I sit within CDRH's Office of In Vitro Diagnostics, or OHT7, on the personalized medicine team. And today I'm going to be providing some information on FDA's review of pharmacogenetic in vitro diagnostic devices, as well as an overview of FDA's regulation of IVDs, specifically looking at OHT7's activities, the pre-market review of IVDs, so demonstrating analytical and clinical performance, and the investigational device exemption regulation. And then I will turn to specific considerations for pharmacogenetic tests, including intended uses and analytical and clinical validation and labeling, and then go over relevant policies and programs, certain relevant policies and programs for the community before opening it up for questions. So focusing on in vitro diagnostics, or IVDs, per FDA's regulation, these are medical devices that include reagents, instruments, and systems intended for use in the diagnosis of disease or other conditions, including a determination of the state of health in order to cure, mitigate, treat, or prevent diseases or its sequelae. And such products are intended for use in the collection, preparation, and examination of specimens taken from the human body. So things that are or can be IVDs include instruments like next generation sequencing, sequencers, reagents used for laboratory testing, and then specimen collection devices such as blood collection tubes. And IVDs are generally used in clinical labs, at the point of care, such as a doctor's office, or over the counter, so sold directly to consumers. So FDA's Office of In Vitro Diagnostics, or OHT7, is responsible for the total product lifecycle activities for IVDs and serves as FDA's primary source for scientific and medical expertise with regard to safety and effectiveness of IVDs throughout the total product lifecycle. And pre-market activities are just one of the activities that OHT7 is involved in, with others including post-market activities such as monitoring and surveillance, post-market inspections and studies, and then compliance and enforcement actions, the issuance and updating of guidances, including policies pertaining to pharmacogenetic tests, and then providing associated trainings, external engagement and outreach, such as involvement in this collaborative community, but also holding public meetings, webinars, and town halls, and then program development and operations. So this could include performance tracking and data reporting. And so through external engagement and outreach, we are particularly interested in community recommendations for performance specifications and test standardization and harmonization. So acknowledging this community's objectives to improve consensus building in the pharmacogenetic community and to provide evidentiary standards for pharmacogenetic tests. And as I mentioned, today I'm largely going to focus on the pre-market review considerations for pharmacogenetic IVDs, as well as certain policies and resources relevant to STRIPE. But before going into pharmacogenetic test specific considerations, I do want to first go over FDA's general pre-market review of IVDs. And so the risk-based classification of a medical device, including an IVD, determines the regulatory requirements and pre-market process as applicable, as well as the risk-based classification and review of IVDs, which is based on the device's intended use and most often primarily the consequences of an inaccurate or misinterpreted result. So medical devices are classified into class 1, 2, and 3 based on the risk of the device, with regulatory control increasing from class 1 to class 3. And the device classification regulation defines the regulatory requirements for a general device type. So most class 1 devices are exempt from pre-market notification submission. Most class 2 devices require a pre-market notification or 510K or a de novo classification request. And then class 3 devices, which are generally the highest risk device, require pre-market approval. So class 1 low risk devices are generally only subject to general controls. And these entail registration and listing, notification, proper labeling, receipt and processing of complaints, reporting problems to FDA, and good manufacturing practices. And then class 2 and class 3 devices are also subject to these general controls, but class 2 moderate risk devices are also subject to special controls, which are specific requirements for specific device types. And then a pre-market notification submission requirement, or a 510K, where FDA would review data and protocols before a device is marketed. And then for class 3 devices, these are the highest risk classification of devices, and these are subject to a pre-market application submission where FDA reviews data and protocols as well as manufacturing information before the device is marketed. So, as I mentioned, FDA review of IVDs is driven by the intended use of the device and the types of validation studies that are needed to support the use or uses of that test depend on that intended use, which includes the information found in the labeling for that device. And so while there are different regulatory considerations for class 2 versus class 3 devices, for example, pre-market approval inspections for class 3 devices, the types of validation studies needed for these devices are generally the same. However, the specifics of each validation study and the considerations for validation testing might be different. And generally, the result to be validated is whatever the final output of the test is. And this could be a single biomarker, or it could be a composite test score for multiple biomarkers determined by an algorithm. And so things to consider when designing and conducting validation studies include the purpose of the test. So if it's intended to diagnose a disease, predict risk, identify a population for whom a drug is effective, the disease or condition the device is intended to diagnose or predict the risk of. This can also be the therapeutic response that's being predicted, the test method, including the specimen types, if it's NGS, using whole blood, or saliva, the patient population. So we'd consider if it's an asymptomatic patient population, pediatric or adult, and then the context of use. So looking at whether the test is performed in a lab, at the point of care, or at home. So the approval standard for medical devices is a reasonable assurance of safety and effectiveness, which for most IVDs generally means a reasonable assurance of analytical and clinical validity for the test's intended use. Again, considering the risk of an inaccurate or misinterpreted results. So for class 3 PMA devices and then novel devices for which de novo classification is appropriate, meaning that it's reasonably likely that general and special controls would be sufficient to assure the safety and effectiveness, FDA would evaluate if the device is safe and effective for the intended use. But for 510K devices, FDA does evaluate whether the new device is substantially equivalent to a legally marketed predicate device, such that the new device is as safe and effective as the predicate device. And so for IVD analytical performance, we generally look at the precision or repeatability and reproducibility. So this is asking, will I get the same result in repeated tests over time, looking at the same day, same operator, same instrument, and same lot? And then for reproducibility, we'd be looking at whether you would get the same result as someone else testing the same sample. So looking at reproducibility between runs, days, sites, operators, instruments, and lots. We also look at accuracy, which is, will I get results that are the same as truth? And truth could be a legally marketed predicate device, a reference method, or a clinical endpoint. We look at limit of detection, which for genetic tests, this is generally DNA input studies to determine the limits of DNA concentration that reliably produce accurate results. We also look at potential interferences or cross-reactivity to inform, what are the limitations of the device, what are endogenous or exogenous substances that could interfere with the results, what factors influence accuracy, when should my device not be used, and then we look at sample stability. So how should samples be collected, handled, and stored to achieve the labeled performance? So generally, clinical validation demonstrates that analytes are clinically meaningful as claimed in the intended use using valid scientific evidence. So valid scientific evidence could be from well-controlled investigations, partially controlled studies, studies and objective trials without matched controls, which include the testing of banked human specimens, well-documented case histories conducted by qualified experts, and then reports of significant human experience with a marketed device, such as in certain clinical practice guidelines, from which it can be fairly and responsibly concluded by qualified experts that there's a reasonable assurance of safety and effectiveness. So generally, clinical performance of a device is established in the population in which the test will be used, which is the intended use population studies. And clinical validation studies should investigate the intended use of that test in the claimed clinical population using the final analytically validated test configuration. And then test results should also be compared to a known truth, which, as I mentioned, might be a predicate device, a reference method, clinical diagnosis, or outcome. And then the inclusion and exclusion criteria, as well as the statistical analysis plan, including performance goals, should generally be pre-specified for clinical validation. So, for example, by defining the performance of the test around a clinically relevant cutoff, if applicable. So that is what we see most at FDA. However, this does, again, depend on the intended use of the test. For example, clinical cutoffs might not be relevant for a continuous marker, for example. But either way, clinical interpretation should be pre-specified, locked down, and then generally confirmed in an independent data set to avoid overestimating the performance of the test. And clinical studies to validate biomarker tests often do have unique statistical challenges. And so pre-specified clinical and statistical analysis plans are crucial here. And I'll go more into pharmacogenetic test considerations with respect to clinical validation later. So I did want to briefly touch on the investigational device exemption, or IDE regulation, which sets forth the regulatory requirements for investigational devices, which are devices that are the object of a clinical investigation and that have not been authorized for the intended use identified in that study. So even if the purpose of the investigation is not to inform a marketing submission for the device, the use of the device in the context of the investigation is generally considered to be investigational and subject to the IDE regulation, unless that device is legally marketed for that intended use. So IDE requirements, including the need for an IDE application, depend on the risk posed to study subjects by use of that specific investigational device in the investigation. And only significant risk studies require an approved IDE application prior to the study initiation. Those studies with non-significant risk devices must still comply with certain abbreviated requirements pertaining to labeling, IRB approval, informed consent, monitoring, records and reports, and there's some prohibitions against promotion. The study risk determination for use of an investigational pharmacogenetic test, again, depends on the risk posed by study subjects by the specific use of that test. So there might be scenarios where a study involving an investigational pharmacogenetic test is exempt, non-significant risk, or significant risk. The takeaway here really is that the study risk determination is not one size fits all. And study sponsors are responsible for making study risk determinations to determine the risk of their test used in the study with their IRB, though FDA can assist with study risk determinations if needed and requested. So moving from FDA's kind of general review of IVDs to more pharmacogenetic test-specific considerations with respect to intended use and validation. So and this is probably familiar with the community, but we generally consider pharmacogenetic tests to mostly be those that are intended to provide information that may aid in the selection of certain therapeutics, or in some cases that may aid in dosage selection. However, it's important to consider that there are various uses and many potential patients to be tested with pharmacogenetic tests. Phenotype is usually not obvious prior to treatment. So, if you're a poor metabolizer or a differential responder to certain therapies, there are wide population differences in alleles and frequencies, and then rare allele combinations can be challenging to validate. And so the types of validation studies that are needed for pharmacogenetic tests, again, as I mentioned many times, depend on the intended use, and different uses have different benefit risk profiles. So for pharmacogenetic IVDs, these various uses can range from tests that only provide metabolizer or transporter phenotype information with no drug-specific information, to those that do provide specific information on a drug safety, efficacy, or dosing. For example, FDA's authorized CYP2D6 assays that are intended to provide just general metabolizer phenotype information for the purpose of providing information to aid in drug selection without reference to a particular drug, as well as CYP2C19 assays that may indicate if a user is at increased risk of citalopram side effects or that clopidogrel is less likely to be effective. So there is a range, and it's also to consider whether pharmacogenetic testing is essential for the safe and effective use of a corresponding therapeutic product. For example, FDA has authorized a POMC PCSK1 LEPR companion diagnostic panel to select patients for treatment with setmelanotide. And in that case, that test was deemed essential for safe and effective use of the drug. So pharmacogenetic assays that FDA has authorized to date generally fall under two Class 2 regulations, each classifying a different generic device type based on the original test submission's intended use and benefit risk profile. So the first classification regulation on the left established was for drug metabolizing genotyping systems, which are intended for use in testing DNA to identify the presence or absence of human genotypic markers encoding a drug metabolizing enzyme or DME to aid in determining treatment choice and individualizing treatment dose for therapeutics that are metabolized primarily by the specific enzyme about which the system provides genotyping information. And so under this regulation, we have authorized CYP2D6, CYP2C19, and UGT1A1 IVDs for general use, meaning the authorization doesn't include claims for specific drugs, as well as VCOR-C1 and CYP2C9 warfarin sensitivity tests. We have also authorized pharmacogenetic tests under a subsequent regulation, pharmacogenetic assessment systems, which are for in vitro molecular diagnostic systems intended to detect nucleic acid variants isolated from human specimens for the purpose of assessing the presence of genetic variants that not only impact metabolism, but also exposure response, risk of adverse events, dosing, or mechanisms of prescription or over-the-counter medications. And so under this regulation, we've authorized an over-the-counter multi-gene IVD with a general use for detecting certain genetic variants that may be associated with medication metabolism, but no drug specificity, as well as more specific uses for providing drug-specific information on citalopram, clopidogrel, and simvastatin. And this regulation is not specific to the detection of variants, including a drug metabolizing enzyme, and the special controls or regulatory requirements differ between these two regulations based on the uses of the IVDs that were classified under them. And so before going into pharmacogenetic IVD validation considerations, I did just want to show some of the different intended uses authorized under the affirmation pharmacogenetic regulations. Again, given that the types of validation depend on the intended use. And these are just select quotes from FDA's public decision summaries for certain pharmacogenetic assays. And, as I've mentioned, demonstrate, tests with a general use that provide a patient's genotype, but don't necessarily provide the association between the detected variant and specific therapeutics. So for example, the Genomatics Cube CYP2C19 system that can be used to aid clinicians in determining therapeutic strategy for drugs metabolized by the CYP2C19 gene product. But again, not to predict drug response, but then also IVDs that provide drug-specific information, for example, the e-sensor warfarin sensitivity saliva test, which again aids in the identification of patients at risk for increased warfarin sensitivity, or the 23andMe Personal Genome Service pharmacogenetic reports for CYP2C19, which provide interpretive drug information regarding the effect of the identified metabolizer phenotype on citalopram and clopidogrel therapy. So really this slide is just to highlight that there are a range of pharmacogenetic test uses for tests that have been cleared by FDA, and also to highlight that there's not, one size fits all approach to pharmacogenetic IVD uses, as well as their review and their clearance. So there are various analytical considerations for pharmacogenetic assay validation and demonstrating analytical performance. So generally, test developers should demonstrate the IVD's ability to accurately and reproducibly differentiate genotypes, alleles, or variants using both the lowest and the highest nucleic acid input concentrations recommended in the product labeling. And you also want to consider what uses and critical alleles that the test includes to establish that there's adequate accuracy estimates for those alleles. Samples should generally be native samples from the intended use population whenever possible, though when fresh samples for rare alleles or genotypes are scarce, we do consider the use of archived or retrospective samples. And then though natural samples are preferred, we also do consider contrived samples, especially for rare variants. We also recommend evaluation of test samples that encompass all genotypes in the test system. And studies should be conducted in the intended use environment. So again, clinical lab, point of care, according to the instructions for use, and should include all steps from sample collection to the test result. And then as this community likely knows, rare variants may pose technical challenges when evaluating pharmacogenetic test analytical performance, especially if not detected by a test. For example, rare variants could prevent primer binding in subsequent test results, or often, with the general assumption that a *1 call means wild type, rare variants could occur that the test doesn't detect and impact allele definition. And there are also instances where one SNP might occur in more than one star allele, and it's unclear what allele is being called for a gene. Intended use is also an important, in informing whether the error rate of a test is acceptable, especially given that what's observed in analytical performance data will inform any necessary limitation or warning statements for the test. It is also generally important where applicable for pharmacogenetic tests to evaluate potential cross-contamination, particularly studies to characterize potential carryover by alternating specimens of known genotype, and whether this has been adequately mitigated, for example, through appropriate validation studies. And then we also recommend characterizing the effects, as I mentioned earlier, of potentially interfering substances relevant to the intended use specimen type, as well as potentially interfering genetic sequences such as homologous regions or pseudogenes on assay performance. For example, for serum or whole blood-based pharmacogenetic tests, developers typically evaluate triglycerides, bilirubin, and hemoglobin as potential interference. And then for saliva-based pharmacogenetic tests, developers typically evaluate, the impact of mouthwash, smoking, toothpaste, whole blood, and eating and drinking on the performance of the assay. So now I'm going to walk through some general considerations for the clinical validation of pharmacogenetic IVDs. So to demonstrate clinical validation of the test, you would need to consider first, what is the relationship between the genotype and enzyme phenotype? For example, which genotypes are poor metabolizers? And then the data and evidence available or needed to demonstrate this, given potential variability in genotype to phenotype translation methods, different interpretations of genetic variants, and then the continued emergence of new information or research supporting rare or less studied variants that could impact existing star allele nomenclature and the assignment of function to alleles. You also need to consider the relationship between that enzyme phenotype and then any drug-specific intended use if included by your test. For example, whether a poor metabolizer is at greater risk of side effects. And then for tests that provide information about specific drugs, during the pre-market review of an IVD, we work with our colleagues in the Center for Drug Evaluation and Research, or CDER, to evaluate whether the pharmacogenetic associations and recommendations provided by a test may impact the safety and effectiveness of the drug or drugs. So I want to highlight cross-center collaboration is very important here. And then there are various types of supporting data and information to establish a test's clinical validity. Again, here, it's not one size fits all is what I kind of want to emphasize. So examples of information or data that can support clinical validation include information from peer-reviewed published studies demonstrating a relationship between the genetic test result and the clinical presentation, drug labeling, or clinical data. And so this could be a prospective study, an analysis of a retrospective study using bank samples. I did include on the slide just a screenshot of a decision summary from a test cleared under the Pharmacogenetic Assessment System regulation to show, where data and literature was used, not just to support the predicted transporter phenotype, but also the resulting likelihood of side effects from simvastatin. This was for an SLCO1B1 test report. So where evidence of the effectiveness of the biomarker is being leveraged from peer-reviewed literature, information to sufficient to demonstrate that the data can be leveraged to support clinical validity should be provided to support that intended use. So here you would consider how the test is used, for example, correlating detected alleles with predicted enzyme activity and impact on drug metabolism versus informing treatment selection or dose adjustment. And then more specifically, how the device used in the published study is relevant to the new device. For example, is it the same version of the test? Or how can comparability between the new device and the device used in the publication be established in order to ensure that the data from the publication can be confidently extrapolated to support the clinical validation of your IVD? And so I do want to also add that IVDs for consumer use or over-the-counter use, including pharmacogenetic IVDs, generally have additional studies conducted to generate data demonstrating that the tests are accurate in the hands of lay users, including sample collection, and that lay users can understand the results. These additional studies are generally user comprehension studies, and we also consider human factors during pre-market review where applicable. But on this slide is an example decision summary excerpt, again, describing a user comprehension study that was required for clearance of a direct-to-consumer pharmacogenetic assessment system pursuant to that device regulation's special controls. So as I mentioned, special controls are tailored to specific device types, and in this case, a user comprehension study was required for authorization. So when it comes to pharmacogenetic test labeling, we generally look at adequate instructions for use, the test's intended use, directions for use, warnings, limitations, interpretation of results, and then a performance summary. And so for pharmacogenetic IVDs, labeling considerations include adequate explanations of associations between detected variants and predicted enzyme function, and if applicable, any specific therapeutic. There are instances where it's important to include specific pharmacogenetic test considerations by race and ethnicity, for example, allele prevalence according to race and ethnicity, as well as relevant variants included or not included in a test, for example, that may contribute to false negative results, but are variants that have different frequencies among underrepresented patient populations or that vary by different ancestries. Limitations and warning statements in labeling are crucial to ensure the safe and effective use of the test so that users can understand the limitations based on the supporting validation, performance, and intended use. And so in cases where an FDA authorized test has certain limitations, these labeling mitigations were informed by and deemed necessary based on the validation that was provided, or in some cases based on the lack of validation that was provided to support use of that test. And so in considering what limitations might be needed, one generally can consider the validation provided and any bias in accuracy estimates. So for example, positive selection bias, you would consider the sample size and margin of error for certain genotypes, for example, wide confidence intervals from low sample number. And then you'd also consider sample types used. So, was validation established using contrived or non-intended use samples? And examples of limitations include when a device does not measure all possible variants, alleles, or genotypes, or when the range of variants, alleles, or genotypes is not known. And so specifics might include interrogated targets such as genes or types of variants the test cannot detect with validated performance, or failure to detect certain rearrangements or indels larger than a certain size or in specific genes. Also the presence of other variants, specifically rare variants that might result in false positive or false negative results. Okay, and so now turning to potentially relevant policies, it's important to understand companion diagnostic policy and whether a test is a companion diagnostic when thinking about pharmacogenetic testing. So in guidance finalized in 2014 by FDA, FDA defines a companion diagnostic as an in vitro diagnostic that provides information that is essential for the safe and effective use of a corresponding therapeutic product. So companion diagnostics often identify patients who are most likely to benefit from a therapeutic product. They can also be used to identify patients likely to be at increased risk for serious adverse reactions. They also can be used to identify patients in which the population for whom the drug was studied is identified and found to be safe and effective. I also want to note the use of a companion diagnostic with a therapeutic product is generally stipulated in the instructions for use in the labeling of both the diagnostic device and the corresponding therapeutic product. So that's an important part there. But for this community, what I want to highlight is that while we consider pharmacogenetic tests to be mostly those that are intended to provide information that might aid in the selection of certain therapeutics, or in some cases that may aid in dosage selection, companion diagnostics are a specific type of pharmacogenetic test where, again, companion diagnostic tests are those that are essential for the safe and effective use of a corresponding therapeutic product. And in general, they've had their own regulations that are different than the general pharmacogenetic regulations that I went through. And then before I switch slides, the other thing I want to note is that the decision regarding whether an IVD is essential for the safe and effective use of a corresponding therapeutic product is made by the Therapeutic Product Center in consultation with CDRH. So another guidance that this community should be aware of is FDA's final guidance clinical decision support software, which was finalized in 2022. At a high level, this guidance describes or clarifies the scope of FDA's oversight of clinical decision support software intended for healthcare professionals as devices, and it describes the CDS software functions that don't meet the device definition. Many pharmacogenetic tests often have device software functions, and if you have any questions regarding FDA oversight of software, I have included the Digital Health Center of Excellence email on this slide. Okay, and so if you have specific questions, particularly about a pharmacogenetic test validation or regulatory pathway, we do encourage you to talk to us using our Q submission process or Q-subs, which is a way for sponsors to meet with FDA for discussions, advice, and formal feedback. This is a voluntary program, and it is a great way to get FDA's feedback before initiating a submission, conducting a clinical study, or even analytical validation of an assay. And this process is part of our FDA's efforts to increase efficiency and transparency for sponsors. And again, it provides the opportunity to address new scientific and regulatory issues. I do also want to note with the QSub program, a sponsor can also obtain a formal risk determination from FDA by submitting a study risk determination QSub. And so this pertains to, determining the risk of an investigational device in a study to see if an IDE application is needed. I do also want to note and acknowledge that the considerations that I've provided today have been general. And they've been purposely general. As this community knows, it's known that each testing system will have a unique set of concerns, and we expect to identify and discuss these unique concerns with individual test developers, for example, through a Q submission meeting. So I do want to acknowledge that, and again, just really highlight this pathway as the mechanism by which you can get input on questions pertaining to pharmacogenetic tests. And I want to highlight, again, as I've had some screenshots throughout today, that CDRH does publish decision summaries for authorized devices. And here I've included the 510K database for reference, as well as FDA's list of nucleic acid-based tests. I should note that the slides will be shared with this group. I also want to note that we have an FDA table of pharmacogenetic associations, which the CDER liaison, Dr. Robert Schuck of this community, previously presented on. Just as a reminder, this table includes associations that FDA has evaluated and believes there to be sufficient scientific evidence to suggest that subgroups of patients with certain genetic variants or genetic variant inferred phenotypes are likely to have altered drug metabolism, and in certain cases, differential therapeutic effects, including differences in risks of adverse events. Again, the table is broken out, as Bob previously presented, into 3 sections. So pharmacogenetic associations for which data support therapeutic management recommendations, associations for which data indicate a potential impact on safety or response, and then associations for which the data demonstrate a potential impact on pharmacokinetic properties only. And I'll note that we do update this table periodically. And I also want to note that the table has an open docket for stakeholders to offer specific comments on pharmacogenetic associations that FDA should or should not include in this table. And then on this slide, I've also included CDRH's Digital Health Policy Navigator, which is a tool to help in determining whether a product's software functions or potentially the focus of FDA's oversight. And then I've also linked the guidances discussed and relevant to today's presentation. And so to summarize the key takeaways from today, CDRH is committed to facilitating access to high quality, safe and effective pharmacogenetic IVDs. And we do strongly believe that community engagement can support consensus building, harmonization and standardization, and are happy to be able to participate in this community. Devices that provide pharmacogenetic information might have various uses that span a continuum of complexity and risk. So there's really no one-size-fits-all approach, as again, regulatory oversight is risk-based and dependent on the test use and the benefit-risk profile. And then lastly, as I went through, there are analytical, clinical, and labeling considerations to keep in mind for pharmacogenetic tests. And we really encourage you to come talk to us using our Q submission program. And with that, I will open it up for questions. I know that there was another agenda item after me, so I'll defer to Ben and Sarah how long I have. But and I'm just seeing the chat now. So yes,
Sure.
let me see.
So it looks like we have time for a few questions before jumping over to Terry Klein. And again, everybody, if we didn't get a chance to get your question answered, McKenna's contact info as well as a copy of the slides will be provided to all members. So you will be able to have your questions submitted and get a response. So kind of going back and slimming up through the chat, McKenna, looks like we had one question come from Don Rule specifically regarding, are companion diagnostics always going to be class 3?
Yeah, so no, we actually have some class 2 authorized companion diagnostics. The one that I mentioned earlier, the POMC PCSK1 LAPR panel is a class 2 companion diagnostic.
Okay. Another question we'd had came from Elizabeth Big. Who defines essential, and would DPYD genotyping be considered essential companion testing, even though the FDA doesn't have labeling that requires this testing prior to treatment with 5-FU chemo?
So as I mentioned, the decision of whether a test is essential for the safe and effective use of a therapeutic product is generally determined by the Therapeutic Product Center in consultation with CDRH. I won't be speaking to specific decisions at this time, but with respect to companion diagnostics, again, it's generally done during therapeutic product review. However, there are instances where you identify signals post-market, and again, FDA will review the data, consider test availability, and make a decision with respect to updating the therapeutic product labeling at the time. I would defer to my colleagues at CDER with respect to additional information on drug labeling. Okay,
another question we had from Josiah Allen was, when can we expect a statement from the FDA that provides an actionable definition of sufficient evidence?
Again, I won't be able to speak to any future activities if and when they're planned. As I mentioned, participation in the collaborative community, I can really only share information that is available to the general public. So won't be able to speak to that, but again, appreciate the questions.
Okay. Thank you again, McKenna, for sharing your time and some awesome information with us today. We'll get copies of the slides out to everybody, but thanks again for joining us today in this all members meeting.
Thanks. And I want to add one follow-up to the question of when can we expect a statement from FDA that provides an actionable definition of sufficient evidence. I think one thing again to highlight here is what constitutes sufficient evidence will depend on a specific use of a test. I know that, it's important to develop community recommendations, policies, and the like, but when, as I mentioned today, during pre-market review of IVDs, there are a lot of unique considerations to a specific test, what that test's intended use is, who the patient population is. So I do just want to highlight, there's not always a one-size-fits-all approach with respect to what's deemed sufficient evidence given the variable uses, clinical conditions, associated therapeutics, and their risk profiles. So just to follow up on that. Thank you.
Okay, thanks, McKenna. With that, I'm going to turn it over to Terry Klein, and we'll let Terry take it from there.
Great, thank you. So this should be pretty quick. For those of you who don't know, and those of you who do know, there's a new association called the American Pharmacogenomics Association. It has an abbreviation, capital A, capital P, capital G, small x, capital A. And there have been a lot of discussions between the members of the Pharmacogenomics Research Network, people that we get inquiries at PharmGKB, as well as CPIC as to what is this association. It's popped up in the last 6 months. I'm sure that many of you have had lots of email that personally I call spam, but email sent from them. And so we did some investigation of it because none of us had been contacted about this association. It just sort of popped up. And so we started doing some investigation work about who they were, what they were putting out, et cetera. And a number of us became concerned about the information that was being disseminated by them. And a lot of that information is, interestingly enough, AI generated. And I find that interesting because I do a lot of work in that area. But we were concerned about the content that is. So for example, there was a summary put out on DPYD, which has some very introductory material, and it mentions a paper from 2011. Yet those of us in this field know that there has been a lot of work moved forward on DPYD testing, et cetera, and newer guidelines since 2011. And so we are concerned about the content of the information that is being disseminated, how it's used, and what this organization is. And so because we've been contacted by many members of these R groups, et cetera, they are looking for guidance after having been contacted directly by that association. And we have decided that we have concerns about this organization. And we have put out a statement of which I reached out to Ben and Sarah on behalf of PharmGKB, the PGRN, and CPIC to see if Stripe was interested in joining the statement as well. We feel that having the major PGX groups represented would be good. We have moved ahead on without Stripe because we keep getting these contents. If you go to pgrn.org, you can see it on their site. But the website statement that we ultimately agreed to was the Pharmacogenomics Global Research Network, PGRN, the Clinical Pharmacogenomics Implementation Consortium, CPIC, and the Pharmacogenomics Knowledge Base, PharmGKB, are not associated with nor endorse the new organization titled American Pharmacogenomics Association. We felt there was a need to do this because there is lack of clarity in the community. We're seeing a number of schools of pharmacy join on. They've done a great marketing campaign. I give them a lot of credit. But it seems rather odd that they haven't engaged with any of sort of the established experts, for lack of a better phrase, of that group. And so in raising this idea to Ben and Sarah about whether or not Stripe would be interested in joining in on that statement, the thought process was we should bring it to the Stripe community at large and get some thoughts. So Sarah, I don't know if, I know we only have a couple of minutes. You could go to the pgrn.org site and put it up there so people could really see it. I don't know how you want to proceed if people have thoughts. I guess I didn't think about this. I thought I was going to post it in chat, which I'll do right now. I'll just do it there. So I guess the question is, do people have any thoughts? Sarah and Ben, I don't know how you, I'm going to turn it back to you to move it forward since you're the leaders of this group. And someone's got their hand raised.
Terry, there are people posting in the chat similar to what you have, and hopefully nobody's applying to the job that Josiah just informed us was posted on Indeed for a sales rep. But I, for one, personally agree with putting a statement out. I'll just stop there.
I will tell you that they are now selling a PGX test, which is like, what? Are you kidding? So, yeah.
This is George. If I may say, we should definitely discourage people from joining. Tell them they're actually, as you rightfully mentioned, Terry, it's a spam. As I wrote in the chat, we've been even bullied when I told them that they're no good. They threatened that what I said may lead to legal, actions from their end, which is ridiculous. So I think we should definitely kill it as much as we can. So yeah.
I appreciate that. So we were very careful on the statement. We went through 5 different iterations, and ultimately we went to legal to, we wanted to make sure that none of us would get sued, so I appreciate you saying that. We did have much stronger things to say. But like I said, we just chose not to. This way we can at least alert people that we're conscious of it and we're not endorsing them.
Okay, great.
Should we hold a vote either in the chat or verbally with ayes and nays? Are we allowed to do that, Ben and Sarah, or what is there a?
I don't know. Sarah, you got to take this over, Ben.
Yeah, well, yeah, I mean, feel free to drop any comments in the chat, but I think what we could do is send out following this meeting a link with maybe, a few options on voting, and then people can provide feedback either way.
Yep, absolutely.
Yeah, that would be great. And like I said, this has been posted on the PGRN site. It will be posted shortly on the PharmGKB and CPIC, and all that goes out. It's just we're in the middle of changing our blog at the moment.
So we'll definitely do that. We'll as a follow up to this meeting. We'll send a couple options, like Sarah said, so that we can vote, so that everybody that's involved in the community that wants to take a position, your voices will be heard, and that part of being a community is we can act like a community when it calls for that, of course, right? And I believe that will be, the best path forward.
That'd be great. And I will tell you that and I'm speaking on behalf, not with my Stripe hat, the other hats that I wear, which are too many, that if Stripe decides to join in on this statement, we will adjust the statements on the websites as well to include Stripe in there, which is what we had originally, but took out because we hadn't had this discussion yet. So thank you for the chance to raise this with the group.
Yeah, no, absolutely. Thank you, Terry, for taking the lead on that and bringing everybody up to speed on, some people are aware of it, others aren't, and that may change the way they think about it. And I think, kind of in summary, great meeting today, everybody. We have an action item here that will, give everybody the opportunity to, raise their voice and raise their hand that wants to join in on this, and we can proceed with us, following by the policies of the community. We'll move forward that way without wasting any time. And thanks again for everybody joining us on this Stripe All Members meeting today. Hope you guys have a great rest of your week, and we'll be in touch with talk with all of you soon. Thank you very much again.
Great, thank you.